out of 64, 61 of them say amino acid. 3 of them just mean stop. That's it. This is the end. Right? Ribosome's job is to show the codons in the proper order. What knows the codons and understands AUG means start and methionine or F-methionine is the tRNA with its anticodon arm. So we looked at that last time also. Each tRNA is specific for a specific amino acid. So this one will have its own specific amino acid up there and won't have anything. It's whatever is associated with that anticodon. All right, so let's see how you get from DNA to mRNA. So I put the asterisk there to tell you, hey, that starts 3 to 5. We read our mRNA 5' to 3', and then we go first codon, second codon, third codon, fourth codon, and we translate it into amino acids. I'm going to try to simplify it. It's easy in animation to see it. Okay, I will use the animation. It's hard to put it in words, so I will put it in words for you guys, a simple way of saying it. But most people have it, look at the animation a couple of times and they get it, but then they're like, I don't know how to write it for you. So I'll go over that. Very basic and fast. All right. So first we have your mRNA. All right, so this is your mRNA. 5' end, 3' right there. Somewhere along here is AUG. All right. Small subunit of ribosome. So when mRNA is ready to be translated, small subunit comes and attaches small unit of ribosome comes and first attaches. I'm not going to put 30S or 40S. Same thing is happening to prokaryotes and eukaryotes. So small subunit of the ribosome. The job of the small subunit is to attach to your mRNA and keep it straight. Why? Because it has to read the codons in the proper order. So if this is AUG and then make this a ACU, the next one, I hope that's not a stop codon. All right. If your ribosome is not straight, your mRNA, your ribosome won't see these 2 nucleotides and would say 1, 2, 3, that's the first codon. Misreads your mRNA. So it has to be straight.
Can you repeat what's the job for the small one?
To keep your mRNA straight. Yeah. All right, so the small ribosome does that. While that is happening, when this is straight, the next thing that is going to happen is your first tRNA starts coming towards the small subunit and finds the AUG. So attaches to it. This is what I'm going to do for the tRNA. So attaches UAC and brings methionine with it. When that is happening, the large subunit comes down. The large subunit, if you recall, I said has 3 chambers, like you see up there. when the tRNA attaches to AUG, so first tRNA attaches there, the large subunit starts coming down, joining the small subunit, placing the tRNA, the first tRNA and first codon, in the middle chamber, which we call P. And place the second codon in the chamber we call A. And the third chamber right now is called P. P, think of it as primary. That's not the name of it. It's actually, P site is peptidyl tRNA binding site. A is amino acyl tRNA binding site. But don't worry about it. Just know it as P, A, and E. P, think of it, that's where the first tRNA always sits. A is advancing tRNAs, second tRNA, and so on. So that's the next one. E is where they exit. Once they dump their amino acid in the right location, a tRNA is empty. A tRNA has been used. That empty tRNA has to go into the cytoplasm and recharge itself, meaning with the help of an enzyme, pick up its amino acid so it can keep translating, right? AUG is going to drop its methionine and go off. That AUG tRNA has to pick up methionine again. All right? This is when the complex ribosome mRNA complex is ready to go. Once this happens, the first tRNA is at the P site. Then the second codon shows up. The tRNA associated with this is going to come and sit too. That's going to be UGA, whatever. All right? So P and A will be filled with tRNAs. The second that happens, the second job of the large subunit kicks in. When P and A are filled with their tRNA, the large subunit has an enzymatic activity too. It comes and makes a covalent bond between the amino acid in the P site and amino acid at the A site. Puts the P amino acid on top of the A, because this is amino acid 1, amino acid 2, all right? And breaks the bond there. So breaks the bond there, creates a covalent bond or peptide bond. This one is discharged. Ribosomes moves one codon to the right. This one goes into this place. So next image, if you want to see, it's going to look like this. Now this one is going to have these 2 amino acids on top of that. The amino acid for AUG is empty, is going to go off from here and go to the cytoplasm and be recharged. The third codon is now again at the A site. The third codon comes. So now let me show you in the animation what is happening. I know it's hard to imagine it. I'm going to mute the animation. And again, I have 2 different animations. is it shows it like it's intact. The other animation, which I'm going to show after I go through this word by word, this is huge and simple, so it's easier to use it. But the first is going to be the small ribosome, small subunit is going to attach, the tRNA is going to come, then simultaneously the large subunit is going to come attached. This one is just going to show them attached and then the tRNA coming. So PA, the first, that's the AUG, the first tRNA is going to come, bring its amino acid. A site, the second tRNA comes. Once that happens, this amino acid is now going to come off of the tRNA at the P site, jump on the ones that is at the A site. Do you want me to turn off the light? You're okay? Okay. So, next thing you're going to see right now is, see, it went up, and as that is happening, ribosome moves one codon down, or one codon to the right. The empty tRNA goes to the E site, is going to fall off. The tRNA that was at the A now goes to the P, has the elongating peptide, polypeptide on top of it. All right. So see, this is going to go off. They're explaining this. This is going to go off. A site is ready to accept a new tRNA. That's going to sit there. As soon as that's there, these two now go jump on top of that. Goes out, another one comes. Continues happening until it reaches a stop codon. When it gets to a stop codon, no tRNA is going to sit at the A site because there is no amino acid. If you don't have an amino acid, no tRNA is associated. That gives the ribosome the signal that we are done. And it will separate, your polypeptide goes, and the ribosome goes back and starts with another mRNA. So now let me play this one for you guys.
The information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific...
Upstream means before. Downstream means after. These are terms that we use for DNA.
Specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
What was that chunk again at the end? Is that the
Release factor.
the release
You
factor?
don't need to know it for my class, but basically there is no tRNA that will bind to the A site. And since there is no tRNA, there is actually a specific protein that recognizes those and attaches. And when that protein attaches, ribosome knows this is the end. And it will separate the polypeptide, and that will go fold on itself and make the protein.
And I noticed that it's way easier to look at the video. I'm not gonna lie. It's way easier to look at the video.
Yeah, exactly. All right, so. All right, how do you put it in wording? This is the simplest way. Messenger RNA is ready to be translated. The small subunit of ribosome attaches to the 5' end of mRNA, making sure the mRNA is straight. The first tRNA for start codon attaches to the mRNA. At the same time, the large subunit of ribosome comes and fuses, creating a complex. Placing the first tRNA at the P site. The second codon will be now at the A site. tRNA corresponding to the second codon will come and attach to the complex at the A site. Once P and A are filled with their tRNAs, the large subunit will create a peptide bond between the amino acid that is placed at P and amino acid at the A site, placing the P amino acid on top of the A. Once the bond is created, the peptide bond, the ribosome moves one codon down or to the right, placing the tRNA with 2 amino acids from A, or moving the tRNA with 2 amino acids from A to the P site. The uncharged tRNA will go to A, E, and exit. Again, A site is open for the next tRNA. This process continues until at the A site you will have a stop codon. No tRNA corresponds to the stop codon. Releasing factor comes and allows for the completion of the process. The polypeptide is going to separate. The large subunit, the small subunit are going to separate. The mRNA can be translated again and again with more ribosomes and more tRNA. All right. So again, it's easier to see it than explain it. Come up with a way that it's easier for you to go. This is one example that I used. All right, now I have here, look at the protein synthesis in bacteria. This is why a bacteria can actually replicate every 20 minutes. That's the DNA. This is an actual electron micrograph. So there is DNA up there. Promoter is active, RNA polymerase attaches and starts making mRNA. That mRNA is ready to be processed. So even before the mRNA is completely made by RNA polymerase, ribosomes attach to it and start translating. So that's what it's showing you. That's why how fast they are. But for eukaryotes, that's not the case. Our transcription process is happening in the nucleus. Even after transcription is done, then we have to process the mRNA. We have to remove the introns, stick or splice the exons together. We have to add a 5' cap and a poly A tail. Then that's our mature mRNA. It comes, and then the ribosomes can attach and start translating. Ribosomes for us can be a free-floating ribosome or ribosomes that are on the membrane of rough ER. Okay? One other difference between us and them is the fact that for them, one mRNA can actually produce multiple proteins, multi-protein, but for us, one mRNA is one specific protein. What they do is they make the mRNA, then they make a long polypeptide, and the cell knows and cuts the peptides. And like, for example, from one mRNA, they can actually get 3 proteins. Sometimes they even pack it, so that makes them even do things faster. They can simultaneously, with one mRNA, they can make 3 different proteins. But for us, it's 1-to-1. So these are differences between us and them. And I have an animation for it too. I'll just have you guys look at it if we're done earlier. I'll play it for you guys. So I also have it in the slides, the most important things I want you guys to know about differences between us and them. So which of the following RNAs are both translated and transcribed?